Hi friends, this is Kavya Tendal here. Today we'll see about the mechanism of apoptosis. This programmed cell death. We'll divide the phase into initiation phase, execution phase, at last the removal of apoptotic body. In initiation phase, we have intrinsic phase, extrinsic phase. In intrinsic phase, the mitochondria plays a very important role. Cytochrome C, which is the pro-apoptotic body present inside the mitochondrial membrane, is naturally impermeable to cytosol because of the mitochondrial permeability. This mitochondrial permeability is maintained by BCL2 family of protein, which is basically BAX and BCL-X1 type of proteins. When this protein is damaged by the cytotoxic drugs taken by the patient, or sometimes the high levels of free calcium in the cell, maybe radiation injury to the nucleus, or maybe excess misfolded protein which makes stress over the endoplasmic reticulum, which is called as ER stress, or sometimes there is absent or failure of signaling in the growth factor receptor, in turn activates the mitochondrial membrane and causes injury. It inactivates BH3 protein. Activation of BH3 protein causes activation of BAX and BAC proteins. This in turn makes the permeability of the membrane to lose. And the cytochrome C, a pro-apoptotic body, enters in cytoplasm. It combines with apoptosis activating factor 1, makes a complex called apoptosome. This apoptosome complex forms activation of caspase 9, and the other mitochondrial protein inhibits the inhibitor of apoptosis, thereby the apoptosis takes place. We'll now see the extrinsic initiation phase. In extrinsic initiation phase, every cell exhibits tumor necrosis factor 1. This tumor necrosis factor 1 has something called as a death domain. When the death domain is being activated by Fas L ligand, it is activated, attaches to something called an adapter protein. When this adapter protein is activated, it activates something called caspase 8. Now this caspase 8 is now ready to degrade the DNA. Thereby the initiation phase continues. Now when there is extrinsic and intrinsic initiation phase being there, activates caspase 8, 9, in turn activates caspase 3 and 6, there is cell disintegration takes place. Then what happens is the cell undergoes apoptosis. You should see here the apoptotic bulb. Now phosphatidyl serine, which is present intrinsically, now being expressed extrinsically, which is signaled and sensed by the macrophage. The macrophage enters to that side. This apoptotic cell thus also secretes thrombospondin, which causes macrophage to recruit that side. This macrophage elicits the immune response and starts phagocytosing. The natural antibody and the complement is being over the apoptotic cell. At last what happens is this cell recruits other macrophages and other immune cells and then phagocytose the apoptotic bodies. Thank you. Stay tuned. Bye.